Okay, let's give an entry. Fine. So, in our previous class, we discussed about uh, amino acids synthesis. Okay. Today, we will discuss about peptides. Okay. So, what are peptides? How the peptides are formed, and nomenclature. Okay. So peptide means it will have a N-terminal end on the left hand side and C-terminal end on the right hand side. So N-terminal, when we are naming it as per the IUPAC nomenclature, so first we should take the left hand side N-terminal end, and the suffix should change with INE with YL by changing into INE should replace with the YL until the C terminal end, and the C terminal end written as such. So that is about the nomenclature. So nomenclature of the peptide means you have to write the N-terminal end first. How will you come to know that the N-terminal end? Left hand side of the left hand side is called N-terminal end. Right hand side is called the C-terminal end. Okay. Uh, then N-terminal end is first written, and the suffix INE should replace it with YL until getting a C-terminal end. Once you are the C-terminal end, you have to write as such. So that is about the nomenclature of peptide. And here I am mentioning the two dipeptide. In this dipeptide, this part is called N-terminal end, and this part N-terminal end means alanine. So in that alanine, INE should replace it with YL. So then it's called alanil, and this is a C-terminal end. This we should write as such. So now the name of this compound is called alanil glycine. So if you want to name a tripeptide, here this part is called the N-terminal end, and here N-terminal amino acid is glycine, and here glycine, that glycine INA should replace with YL, and then this part is alanine amino acid. In this alanine amino acid, INE should replace it with YL. So then it is glycyl alanil, and this is a C terminal end, and this part is alanine, phenyl alanine. So now this since it is a C terminal end, we have to retain the name. So that is how the tripeptide named as per the IUPAC nomenclature. And this is a representation in terms of symbols. GLY stands for glycine. ALA stands for alanine. PHE stands for phenylalanine. So this is how one can name the peptides, more particularly dipeptide or tripeptide or pentapeptide or decipeptide or polypeptide. So whenever you want to name it, first you have to look into the N-terminal end. Always N-terminal end is written in the left hand side, and C-terminal end is always written in the right hand side. So when you are naming, you should start with the N-terminal end amino acid, and when you are naming, the suffix ine should replace with yl until getting a C-terminal end. C-terminal end name is retained. This is what the IUPAC nomenclature method of naming the IUPAC nomenclature of peptides. And you know that how the peptide is formed. So peptide is formed by the interaction of amino group of one amino acid with the carboxylic group of the other amino acid. So that is how the peptide is formed. And the obtained the linkage CONH linkage is called peptide bond, and the linkage is called peptide linkage. Okay, this is about the previous class. So now I will go to the next slide. So look here I'm talking about the various possibilities in peptide and peptide structure. Okay, peptide or protein structure. I think you know the difference between the peptide and protein. Anyway, now also, so peptide means what you call. So a peptide is formed by the combination of amino group of one amino acid with the carboxylic group of the another amino acid. That is the peptide. So anyway, I will also talk about the classification. I will also explain what is protein, what is peptide, more particularly polypeptide and pentapeptide when it comes to the definition. So now I am talking about, suppose three amino acids are involved in a peptide bond formation. Okay, here, so there are two, here I am talking about the two amino acids. So two amino acids can combine to form a dipeptide. Three amino acid can combine to form a tripeptide. Four amino acid can combine to form a tetrapeptide. So let's look into how the two amino acid can form a peptide bond. So look into this. This is a glycine. Suppose a amino acid called glycine and alanine. So glycine and alanine can combine can form a peptide. So the possibilities. So here the possibility of peptide formation. So what is that? Carboxylic end of the glycine can react with the amino end of the alanine, gives rise to glycyl alanine. Similarly, carboxylic end of the alanine can react with the amino end of the glycine, can form a alanyl glycine. So what it means, if we are taking a two amino acid, glycine and alanine, it is possible to get a two peptides. Okay, that is glycyl alanine and glycyl alanine and alanyl glycine. So what will happen in the first glycine here what you just look into this structure. So in this structure, this part is called glycine structure and this part is called alanyl structure. So in this always a peptide is written in such a way that N-terminal end is on the left hand side, C-terminal end in the right hand side. So this is N-terminal end, this is C-terminal end. And if you want to name this particular dipeptide, what you can write, so this is N-terminal end, and what is the amino acid? The amino acid is glycine. INE should replace it with YL, glycyl. And this is a C-terminal end, you have to retain the name. This C-terminal end amino acid is alanine. So that is how glycyl alanine. And in this, what you need to recognize, this is the representation in terms of symbol, GLY ALA. It indicates that it's a dipeptide. And in this dipeptide, what you can recognize another thing, glycyl is the N-terminal end amino acid, alanine is the C-terminal end amino acid. And here, what you can recognize, look here, this is alanyl. Alanil means alanine N-terminal end, and C-terminal end is glycine, exactly reverse of this one. So IUPAC name is alanil glycine. So in this, alanine is the N-terminal end and glycine is the C-terminal end. So it is the possibility, if suppose if two amino acids are joined, means two possibilities are there. So for example, glycine and alanine can combine to give a two peptides such as the glycyl alanine and alanyl glycine. So in glycyl alanine, glycine is the N-terminal end. In alanyl glycyl, alanine is the N-terminal end. So this is about the possibility of forming a dipeptide with the two amino acid combination. So with this, I will go to the next slide. So next slide, suppose three amino acids can combine in 6 different way to form a tripeptides, 6 tripeptides. For example, here I am taking glycine, alanine and phenylalanine. Glycine, alanine and phenylalanine may combine to give following tripeptides. So glycine, alanine, phenylalanine, it's one tripeptide. Phenylalanine, glycine, alanine, it's another tripeptide. Phenylalanine, alanine, glycine, it's another tripeptide. Alanine, phenylalanine, glycine is another peptide. Glycine, phenylalanine, alanine is another peptide. Tripeptide, alanine, glycine, phenylalanine. So this is how, without repeating the amino acid, it is possible to get a 6 different tripeptides. Okay, as the size of the peptide increases, the possibility of variation in the structure increases enormously. As the size of the peptide increases, the possibility of variation in the structure increases enormously. So for example, a decapeptide, contains amino acid units is far smaller than a protein. So even then, the number of different decipeptides that can be constructed without repeating each amino acid, okay, even then that can be constructed without repeating each amino acid is around 40,000 crores. Okay, that means without repeating the amino acid with the 10 amino acids itself, it is possible to construct this many number of decipeptides. So, but in the case of protein, amino acids can be repeated. So in that case, it is possible to construct a decipeptide of infinite numbers. So that is about the construction of peptides by using a amino acids. So with this, I will go to the definitions. So what is peptide? So peptide is a short chain of amino acid residues. Peptide is a short chain of amino acid residue that has a defined sequence. Defined sequence means glycyl, alanine, phenylalanine, okay, isovaline, okay, valine, okay, tryptophan. So that is a defined sequence. So will not be changed the order. If there is a change in order, means that will be a different peptide. Okay, that means a peptide is a short chain of amino acid residues that has a defined sequence and generally does not have a fixed three-dimensional structure. Generally, that does not have a three-dimensional structure as far as a peptide is concerned. Suppose a peptide containing 3 to 9 amino acid residues. Suppose a peptide containing 3 to 9 amino acid residues is called oligopeptide. So oligopeptides means it is a peptide containing 3 to 9 amino acid residues. So while the one can combine 10 and more residues are called polypeptides. Then what are polypeptide? Polypeptide means these are the peptides containing 10 amino acids and more. So more than 10 amino acids are involved in a peptide bond formation means such peptides are called polypeptides. Suppose a polypeptide, okay, polypeptides with molecular weight, polypeptide with molecular weight more than 10,000. Polypeptide with molecular weight more than 10,000 and having a definite three-dimensional structure are called proteins. So now you know the difference between the peptide, polypeptide and proteins. So peptide means maybe it's a short chain amino acid residue containing peptides are called residues containing ones are called peptides. If it is a 3 to 9 amino acid residue containing ones are called oligopeptides. If it is a more than 10 amino acid residues containing one is called polypeptide, and the polypeptide with more than molecular weight 10,000 more than 10,000 molecular weight and having a definite three-dimensional structures are called proteins. Okay, this is about the definitions of peptides. More particularly, you can also say about the classification: peptides, oligopeptides, polypeptides, and proteins. So with this, I will go to the next slide. So next slide, in the next slide, I will suppose one polypeptide or a peptide or oligopeptide is given to us. Then what we need to do, if we don't know about the sequence of amino acid present in that peptide, so then we have to find out the structure of peptide. How we can do? So there are methods to find out the structure of peptide. So that we have to discuss. That is called determination of structure of peptide. So I will go to the next slide. So determination of structure of peptide. So determination of the structure of a particular peptide involves determination of the structure of particular peptide involves so what amino acid residues make up the peptide and how many there are. That is the one thing we need to know. And the sequence in which the amino acid follow one another. The sequence in which the amino acids follow one another along the chain. This structural determination is carried out in three ways. So one is the first one is complete hydrolysis. So suppose a peptide is given means that is was taken and it was treated with the HCl, more particularly dilute HCl. So what will happen? All the peptide linkage present in the peptide cleaves and giving rise to amino acids. Okay. So complete hydrolysis means the peptide is replexed with the dilute HCl so that all the peptide linkages are broken. Analysis of the resulting solution reveals the kind and amount of amino acid present in the peptide. But this hydrolysis will not give you any idea about the sequence of amino acid in the peptides. So what it means? Suppose a peptide is taken and treated with dilute HCl and it is reflexed. So what will happen? All the peptides present in a peptide cleaves and giving rise to amino acids. So by taking that solution, if we are analyzing that, we will come to know that what are the amino acids are present and how many number of the amino acids are present, but not the sequence, the order in which they form peptide. So is it in the glycine, alanine, phenylalanine, or phenylalanine, glycine, alanine? So that order or sequence we cannot come out with this complete hydrolysis method. So complete hydrolysis method, it's only useful for finding out the amount, means quantity of amino acids are present in that peptide. Okay, this is about complete hydrolysis. And one more method is called Sanger's method. So second method is called Sanger's method. Okay, Sanger is a scientist, more particularly Frederick Sanger. So he is from Cambridge University. So he introduced this method in 1945. So more particularly, based on this, one can frame a question. The question is, what is Sanger reagent? And mention its importance in the determination of the peptide structure. So this is the 4 marks question or 5 marks question. So what is Sanger reagent? And mention its importance in the determination of peptide structure. So here, Frederick Sanger of Cambridge University, he introduced this method in 1945, and the reagent is dinitro. Dinitro means this is one nitro, this is another nitro. So dinitro fluoro, this is a fluoro. Okay, dinitro fluorobenzene. This is the Sanger's reagent. So what will happen here? A amino group, okay, the lone pair present on the nitrogen can attack this carbonyl carbon, this carbon, benzene carbon. And I think here the nucleophilic substitution reaction. So nucleophilic substitution reaction means lone pair present on this nitrogen attack this aromatic carbon and fluorine will go out. And this moiety is called a peptide. And we know that always a peptide contains left hand side is the free N terminal end, and the right hand side is free C terminal end. So what it means is a peptide is taken, the peptide was treated with Sanger's reagent, and the Sanger reagent is called dinitrofluorobenzene. So what will happen? The free amino group of the N terminal peptide reacts with dinitrofluorobenzene, gives a substituted product. Okay, so this substituted product subjected to hydrolysis. This substituted product subjected to hydrolysis. So when there is a hydrolysis occurs at the peptide linkage. So hydrolysis occurs at the peptide linkage. So what it will form? It will form a amino acid, components of amino acid is obtained. In addition to that, one more compound is also obtained. The compound name is called DNP derivative. DNP means dinitrophenyl derivative, means amino acid derivative. So what it means? So it means that DNP reacts with the N-terminal amino acid. On hydrolysis, DNP intact with the N-terminal amino acid. So by this method, it is possible to find. By this method, it is possible to find N-terminal end of the peptide. So we will come to know that only what is the amino acid present in the N-terminal end of the peptide. So that is the result. And this method, it is not useful to find out the sequence. So the sequence means the order, okay, in which order, means phenyl, alanine, glycine, serine, the order or sequence is not possible to find out by this method, because so why it is, because when the substituted peptide is hydrolyzed, all the components, okay, all the peptide linkages are hydrolyzed. So that is the reason Sanger's method is only useful to find out the N-terminal end of the amino acid, but not for the sequence of the amino acid. So that is the information you need to give under the heading of Sanger's method of determination of structure of peptide. So this reaction you need to write, and explanation also you can give. So what explanation you can give? Frederick Sanger in 1945 he used dinitrofluorobenzene to find out the N-terminal end of the peptide. So what will happen when a peptide is react with the Sanger's reagent? It will give you substituted product. This substituted product on hydrolysis, all the peptide bonds present in a substituted peptide breaks, forming a component of amino acid. In addition to this, a DNP derivative is obtained, and the DNP derivative is the colored compound, and it can be easily isolate. So by this way, it is possible to find out the N-terminal end of the amino acid, but not the sequence of the amino acid present in a peptide. So this is about Sanger's method of determination of a peptide. And one more method is there. It's called Edman method. So that I will discuss in the next slide. So this is Edman method. So here, so one can frame on this question, how is Edman degradation? So this is the question based on this Edman experiment. How is or what is Edman degradation? How it is useful in the determination of amino acid sequence in polypeptide? Okay. What is Edman degradation? How it is useful in the determination of amino acid sequence in polypeptide? This is the question. So more particularly, this for 5 marks question or 4 marks question. So from these two methods, Edman method and Sanger's method, one question is sure for the examination. So what you need, what is the Edman method and what is the Edman reagent? So that we need to know. So look here, this is the Edman reagent. So what is the name of this Edman reagent? So you can look into that. This is the phenyl group, and here nitrogen, carbon, nitrogen, double bond carbon and double bond S. It's called phenyl isothiocyanate. CYANATE. Okay, phenyl isothiocyanate. Okay, this is the Edman reagent. So what will happen? A Edman reagent when it is treated with a peptide. So you know that peptide means it will have a N-terminal end. So N-terminal end of the peptide react with the phenyl isothiocyanate, forming substituted thiourea. So look here, NHC double bond S, NH. Okay, this is called thiourea. Since there is a substituted thiourea is obtained. How the substituted thiourea is obtained? It is obtained by the reaction of the peptide and the Edman reagent. So obtained substituted thiourea undergoes a mild hydrolyzing agent. So that means, so this substituted thiourea, when it is subjected to mild hydrolyzing agent, so that means in a it does not require drastic condition. So mild hydrolyzing agent itself converts this particular converts into a phenyl thiourea identine. Phenyl thiourea identine and one amino acid shortened peptide. One amino acid shortened peptide. So this will be this can be easily isolated. Phenyl identyne can be isolated. So phenyl identyne thiourea identyne contains N-terminal end of the amino acid present in this peptide. And here shortened peptide, it will have all the amino acids are intact in the shortened peptide. So what it means? So this shortened peptide can be separated, and this can be further treated with the Edman reagent. So thereby we'll come to know that one more amino acid present in the shortened amino acid. Amino means shortened peptide. So again phenyl thiourea identyne is formed. So that was identified. Then again it was treated with the Edman reagent. So what it means? It means that Edman method is useful for finding out a sequence of amino acids present in a peptide. So that is the advantage about Edman method. So what is Edman reagent? Edman reagent is phenyl isothiocyanate. So what is the process? The process is phenyl isothiourea. It is treated with peptide. So it will form a bond with the N-terminal end of the peptide, and the obtained compound is called substituted thiourea. Substituted thiourea, when it is treated with a mild hydrolyzing agent, N-terminal end of the amino acid forms a phenyl thiourea identyne, and in addition to this product, one more product is obtained, is shortened one amino acid shortened peptide is obtained. This one amino acid shortened peptide is further treated with Edman reagent to find out the sequence of amino acid present in this peptide. So this is about the Edman reagent. Overall, you need to remember that Edman reagent is useful for finding out the sequence of amino acid present in a peptide. So that is the word you need to remember. You need to remember about the reagent, and you need to able to write the structure of peptide. You need to able to write the structure of substituted thiourea, and when this is hydrolyzed, more particularly it will hydrolyze a peptide bond, and one amino acid converted as a identyne. So if you want to know about this, look here. So there will be a breakage here. So if there is a breakage here, there will be a new bond formation between this nitrogen and this carbonyl group, and forming a identyne, and this part is a shortened peptide. So after the separation of this, shortened peptide was taken, and it was further treated with one more equivalent of Edman reagent to find out the N-terminal end of the shortened peptide. So again, that was treated with one more Edman reagent. Like that, one can find out the sequence of amino acid present in a peptide. So this is about another method of find determination of structure of peptide by using Edman reagent. Okay, now tell me, do you have any doubt up to this? So that means Sanger's reagent and Edman reagent. So complete hydrolysis means all the peptide linkages are giving breaked, and only that will give you the amount of amino acids formed. By the complete hydrolysis, only the amount and number of amino acids are present. That can be concluded by the complete hydrolysis. In Sanger's reagent, N-terminal end of the amino acid can be found out or discovered. Edman reagent means sequence present in the amino acid peptide can be found out. So this is about the determination of structure of peptide. So do you have any doubt in this? If there is no doubt, means I will go for a next concept. No doubt?
No, sir. No, sir.
Okay. Okay. So I will uh, I will take up one more thing. So I think you are aware what is called as cleavage, peptide cleavage. Uh, so this is okay. Maybe what I can do, I can stop at this stage and I will continue in the next class. Okay, why? Because if I'm starting this, I should continue. Uh, it will take maybe 20 minutes or something like that, and there will be a gap 3-4 days. So what I mean to say that the cleavage of peptide bond I want to complete it in a 2 class, so that I will do it in the next class itself. Okay, so what I will do, I will stop at this stage. If you have any doubt, you can ask me. Otherwise, you can log out.